Yeah, okay, welcome back, everyone, and Happy New Year. So today is my pleasure to welcome Islem Rekik, who will be talking about the landscape of generative GNNs in network neuroscience. So this is the sixth talk in the GNN series, and looking forward to your talk, Islam. Yeah, thank you.
Thank you very much for the kind invitation. It's very nice to see that there are many people here joining today's talk. So what I'm going to present today is a brief overview of the landscape of generative GNNs with application to a particular field, which is brain connectivity or network neuroscience. We also call it connectomics. So let me just move this a little bit so I can get my screen clear. All right, cool. Awesome. So the brain can be modeled or represented as a matrix. So this matrix right here, it captures the connectivity weights or different types of interactions between brain regions. So for example, this is a particular brain region in the brain, and we have another brain region here, and the value here reflects, for example, the density of the connections, the number of connections between these two regions. So using this matrix, we can represent the brain as a graph, or also we call it a network or connectome in the field of connectomics, where we model the brain connections and we measure them using different image acquisition tools. So the brain, trying to understand the brain, literally your brain in the matrix, is a very interesting and it could be a complex process to tackle. So because once we understand how to depict patterns within this matrix and across different individuals and subjects, we can predict multiple outcomes, because the brain matrix, it encodes different states. For example, it can tell us if you're feeling sad, depressed, happy, inspired. It also lets us eavesdrop on your decision-making processes, like if you're about to say a yes or a no to a particular question, and sometimes also eavesdrop on your love life. So there are so many things that this simple graph can tell us about who you are and your identity. So your brain in the matrix is your fingerprint, and there has been a lot of studies that showed that the brain connectome or the graph brain fingerprints each one of us. So this is, for example, a beautiful, colorful connectivity matrix and adjacency matrix of the brain graph that shows us the dissimilarity in morphology between different cortical regions. So I'm not going to go into the details, but the idea is that once you build the surface of the brain, the cortex, you can parcellate it or divide it up into smaller regions, and each of these regions will have a particular geometry. So calculating the similarity between the geometries of pairs of regions gives us this, what we call a morphological brain network. So this is a thickness-based. We use the thickness as a morphometric measurement of the brain geometry. And you can see here that for this newborn baby, after 3 months, you can see dramatic changes in the brain, okay? So now trying to also understand the changes in the pattern and even predict them, like see how if we can predict 3 months from 0 months is also a very complex and interesting problem to solve. And here is another matrix. So this is, for example, a different type of connectivity. We call it the functional brain graph or connectivity. And this is of a 75-year-old healthy subject. And for this one, this is a matrix diagnosed with what we call mild cognitive impairment. And you guys can see that there are tremendous or like dramatic changes in the brain pattern connectivity, in the graph connectivity between the healthy state and the disordered state, okay? So trying to understand and analyze these matrices and population of matrices of brain graphs is an important question or important like endeavor because it allows us to gain a deeper understanding, a fundamental understanding of how the brain operates at the macro scale. So here, these connectivity matrices or these graphs, they represent the large-scale interactions between big parts of the brain. But of course, once we are able to map these down accurately, we hope that we're going to get a more microscopic neural level mapping of the brain graph as a highly complex system. So understanding these is very important because it allows us to diagnose subjects, to do some prognosis, also to track, the healthy matrix development. So I'm going to give you a few examples today. So now you may ask, okay, so you got this matrix, but how did you actually get this matrix? So every brain network or graph needs an image. So we're using medical MRI, medical resonance imaging, magnetic resonance imaging to get these networks. So basically we take an MR image and we do some image processing. We pass it through a long image processing pipeline. There are so many software available publicly. And then this image processing pipeline will transform your MRI sequence of the brain into a graph. And that's how we get it. Now, this may be bad news for half of the world because, as you can see in this review paper published by the Lancet Oncology in 2021, we have a big medical imaging gap between the North and the South. So here you can see that, for example, in Africa, in South Asia, there are so many countries with 0 to 2.4 MRI units per million inhabitants, which means, you can in one country, for example, you can have only 1 to 2 MRIs in the whole country. And that wouldn't allow us to collect data or to kind of generate the connectome, because you wouldn't be able to create these brain representations and understand the brain as a graph, examine it as a graph without the MRI. So one mainstream solution to this problem is to, of course, increase equipment, which means install different MRI scanners in these countries, and also having more technical, digital, giving digital training to physicians who use these MRIs. So it's a very costly solution to go with, this basically. So an alternative solution would be maybe trying to think about the problem differently. So here is a question for you. Can we diagnose early and better with minimal resources, which means limited data, less scanning, less resources, and at lower costs? Now, this question is something that I'm really interested in solving. I would say that this is a big question where there are lots of layers and levels that we need to kind of tackle before we get to the stage where we make healthcare and medical imaging and AI with medical imaging affordable, minimalistic. But if we can solve this problem, we will solve it for the whole world. We won't solve it only for a few, countries. So this is a big question here. And if we can partially or fully solve this problem, we can get an input data X. So let's say here, X denotes the data that is affordable to acquire, accessible. And this data is the input data that we want to have. So it's not, it can be of low to middle quality. So we don't need a very high quality data. So it's low quality data. It's easy to acquire. It is accessible. And on the other hand, we have the X hat. So our X hat here is our target data to generate. So X hat would be costly to acquire, inaccessible maybe in many countries, or not conventionally used, even in high-income countries. And ideally, it would be of high quality. So our goal is to learn or to design a GNN model or a deep learning AI model that allows us to get or predict or generate X hat from X, okay? So if we are able to do that, we will solve this kind of problem, which means we'll start from a minimal or partially minimal data, and using lower, low cost, limited environments or limited regimes to acquire the data, and then we're generating the data that is costly to acquire, okay, directly. So going from data to no data, this is the generative path, and here we, over the past few years, we published several works. At the time, back in 2019, we used to call it predictive learning. Now we call it generative learning. But it's the same thing. I mean, there are lots of analogies and similarities, but let me explain where we're from, our starting point. So as I mentioned earlier, we definitely need an MRI scanner for brain connectivity. So there is like the MRI, and there's also the EEG, but EEG hasn't been used in the literature of neuroscience, network neuroscience, to define what we call brain connectomes or brain connectivity, although it's very affordable. So that's a different topic I can talk about another day. But here we're starting from what we still need an MRI. So I'm not solving the problem, but I'm trying to partially solve the problem and alleviate the cost of acquiring more data. So let me explain. So in connectomics, we start with what we call a T1 weighted MRI. So a T1 weighted MRI is a conventionally acquired MR sequence if you're doing imaging of patients, okay? Now, there are more advanced sequences such as functional MRI, resting state, and diffusion MRI. So these require more time. So which means the patient needs to lie in the scanner for maybe 30 minutes and then extra hour to get the other additional multimodal images, the functional and the diffusion. And in traditional brain connectomics or network neuroscience, we have always used the functional and the diffusion to get the graph. So this graph right here was always generated from, these costly sequences. So here in our early works, back to 2018, we introduced the concept or the definition of a morphological brain network that could be derived from simply the conventional sequence. So we circumvent the need of getting fMRI and diffusion, and we try to define the connectome first from this, I would say quite affordable, not totally affordable, but we start with this sequence and we hope to get more costly sequences. So that's our starting point, and we hope, of course, to improve this. So ideally, we would start with an EEG and get to, for example, the MRI-based connectome from the EEG. So that's a different topic. But anyways, so quickly here, so we introduced the concept of a morphological network. Now, our goal is to create or generate directly the X hat. The X hat will be the functional network from this morphological one. So we're trying to do cross-domain generation, diffusion domain, functional domain of the same graph. So the type of connectivity here would change because here it's morphological, but in the other domains, it will mean something completely different. Now, for the resolution also, we try to super resolve this graph by generating finer scale representations of the brain graph. So instead of acquiring the data, we're generating data across resolutions and across time. So basically, in this three-dimensional space, given a single observation of your brain at a particular time point, we are able to span the full space to predict your brain after years, months, to predict different representation of your brain graph across different domains, and to predict also high resolutions of your brain representation of your brain graph across different scales. So basically here, everything becomes generative and learnable, and any point in this space will be generated from a single limited observation. So let me explain this a little bit in a more abstract also way. So these solutions are quite generic. So let's start with the first dimension, which is domain. So graph domain generation. So given a source graph, ideally this graph will be affordable, quite affordable to some extent to acquire. And then our goal is to generate or design a solution, a mapping F that allows us to predict the target graph in a costly domain, okay? So this is high cost data from low cost data. And for example, here, predicting the functional brain graph from the morphological. Now, the multi-graph prediction or generation solves this problem jointly across multiple domains. So given a single source graph, our goal is to learn a mapping that generates all these representations, diverse representations across different target domains. And all of these would be also representations of the brain graph. It could be anything else. So if you're working on, for example, molecular data, biological data, other types of graph data, so this generative model will predict a multi-graph from a single graph. Now, for the resolution, we introduced the concept of graph super resolution. So to the best of our knowledge, the concept of super resolving graphs it wasn't there until we published the first paper back in 2018. And it was about super resolving brain graphs. So in graph theory, generally, we aim to make graphs smaller. So you have huge graphs with millions of nodes. You want to compress graphs. You want to filter graphs. You want to kind of, extract important information from graphs. But, like you don't think about making graphs, bigger and bigger and bigger, like infinitely. So here in this case, we draw inspiration from the field of computer vision, where we do image super resolution. So this problem is very tailored for brain graphs. So let me explain why. So first, to get this graph with only 4 nodes, so let's say you want to divide the brain into different nodes, like for example, 35 regions, okay? Each hemisphere, 35 and 35. So you can use a software, a public software, for example, a very commonly used software is called FreeSurfer. So this FreeSurfer will take the brain image and it will pass it through a processing pipeline and it will output the graph. So that would take probably, I would say like it took 8 hours per subject on average. So 8 hours for each subject to get this graph. So the image processing is very costly. It's also time consuming and resource consuming. If you add more nodes and you want finer parcellations, you're going to spend more time. So here in this graph super resolution learning paradigm, number 1, we circumvent the need of acquiring the image. Number 2, we circumvent the need of processing the image. So we don't need an image in the target domain and we don't need to process it. So we directly generate the high resolution graph from this input one, and this is in the testing stage. So once you're basically testing the model, you're generating this representation in a different domain. You can do it in a different domain. Now the third dimension in our generative space is time. So given a single basically a single observation or a single graph measured at time t0, we want to learn a time-dependent mapping or function f that allows us to transform sequentially the graphs onto follow-up observations and predict the full dynamic trajectory of the graph evolution over time. And this is also, so I'm going to give a few examples later about these. And then there is another concept that we have introduced, which is what we call the connectional brain template. So this concept is an idea where we wanted to show that we can create templates of populations of graphs. So the concept of templates also is prominent in the field of medical imaging, where you have a bunch of images, MRIs, and you just integrate them into a single image, which we call a template that represents that particular population. So it's a data fusion problem. So here, our goal is that to use this kind of graph integration or population-based integration to guide the learning of these generative tasks. So I'll give a few examples later. So here, our goal is to take a single subject, multiple subjects. So each subject here, one subject is represented by different connectivity types. We call them views or data views. So each one is represented by a bunch of matrices, and we have a bunch of subjects. And our goal is to integrate or collapse all of them onto a single matrix, okay, a single graph. And this is called the center graph or the template graph of this population. Now you may ask, okay, so you generated all this data, so what's the point? Why we're doing all this, generative learning? So the goal is actually to use the source data along with the predicted data, and if we trust, of course, the predicted data, to do a disease fingerprint prediction, which means identifying holistic biomarkers across maybe different domains, because here, if you rely on the source, you're limited in your knowledge. If you predict or generate, then you're going to augment your knowledge and you can discover more holistic biomarkers across different domains or different resolutions. And you can do better classification of different diseases, like here, for example, distinguish between disordered subjects and normal controls. So all these works are published and they're available on GitHub YouTube channel. So there are different talks, but There's also a review paper that we published in 2022 on overviewing the majority of these methods, so on graph neural networks. So if you're interested, feel free to check the GitHub repo and also the review paper, which is called Graph Neural Networks and Network Neuroscience. So we also try to be inclusive, promote, reproducible and open science. So the codes, as I mentioned, are available on how to run. If you're interested in running on one of those codes, there is a code demo playlist, and also I think now we have more than 60. So this has not been updated, unfortunately. So sorry, apologies about that. But yeah, so if you're interested also about learning these, the topics I'm going to present today, and you want to go in depth, so you can also watch the oral presentations. So most of these papers are presented, are recorded and presented orally. So yeah, so today I'm going to give you a few examples because we have limited time. So I'm going to brush over the most important idea, and let's start with cross-resolution prediction. So this work was presented by my former PhD student, Islem Rekik, and it was a paper published in one of our top conferences, actually one top conferences, IPME, and selected for oral presentation. So this was the first paper that designed or used a graph neural network for super resolving brain networks, or designed a GNN model for graph super resolution in general. So what is the problem statement? So here, the question that we're asking is how to predict intermodality non-isomorphic brain graphs from a base graph. So basically, we take a graph, an input graph from a particular modality, and we target to generate a higher resolution graph in a completely different domain. So we're solving two problems in one framework. So conventionally, the approaches that have been developed at the time, first, they only handle graphs with the same resolution. And number two, when you have, for example, the orange graphs here, they have a distribution different from the purple graphs, which lie in a completely different domain. Okay, so the problem is that when you try to do this generative task from orange to purple, you're living on completely different spaces, and your prediction or generative power will have a limited capacity. So here what we propose is first we allow for a shift in resolution of the graph, which means we can predict high resolution graphs with resolution nr prime by nr prime from a low resolution graph. And we also solve the problem of what we call domain shift between the source and the target during the learning process. So let me introduce to you Iman GraphNet. So Iman GraphNet has two building blocks that are co-trained together. The first block is an intermodality domain aligner, and the second one is a super resolution block. So here we use a simple GCN with dropout layers and batch normalization. So let me explain the architecture. So first we have our input data. So these are the input adjacency matrices, the brain matrices. So we have like X1S until XNS. So we have our brains. And we add some defined features, of course, there, but I'm not going to go into the details. So the key idea here, using a GCN, a simple graph convolutional network, that acts or is trained as an aligner. So how do we do that? So basically, the hypothesis that we have is that if you have two different data points living in a completely different spaces, one on Earth and one on Mars, and you want to aggregate them, or you want to map one onto the other, the domain shift will create a problem in your learning process because you're not learning in the same space. So the idea is to bring one space closer to the other, and then you do the learning in that shared space where everybody agrees on the same terms. So that's the basic hypothesis here. So that's why we introduced an alignment step, a pre-alignment step. So we have our original graphs passing through a GCN that has a particular loss. The loss function is a KL divergence loss that minimizes the distance between the purple distribution of the source data, the aligned source data, and the target distribution, the blue. So So what we have here is we have the target data, the target data distribution or the target networks. And our goal is to, for every single matrix here, we want to preserve the topology, but we want to shift the distribution, which means the distribution of each brain network, the purple one, after it passes through these layers, it will have a distribution here similar to the target domain. Okay, so each generated X here it has its distribution, source distribution aligned arrow towards the target. Okay, so after this alignment, now we're going to have our source graphs, but they have exactly the same distribution of the target ones, thanks to our loss function. So now we can do the super resolution task in the same space. So basically, here we take these generated aligned graphs, so the source graphs, and we pass them through the intermodality graph GAN. So we use a graph GAN architecture where we introduce an upsampling, a few upsampling layers here. So the model is still, the network or the graph resolution part is still very simplistic. And then basically, as we shift the dimensions, we get our final output matrix of size NR prime by NR prime, which is regularized by the discriminator. So here, basically, this is the super resolution step. And after each of these training samples passes through the network, we get the X hat T1, which is the predicted X matrix for the target domain. And this is subject one. Now, we train this. We have proposed different sub-losses, and please feel free to read the paper if you're interested. But we evaluated this on the SLIM data set, and we super resolved the 150 morphological connectomes of resolution 35 by 35 to 160 and 160, while generating the functional connectivity. So we took the morphological connectivity to low resolution, and we generated a functional connectivity high resolution. And this is costly because functional connectivity is derived from functional MRI. So these are the results. So let me show you the results. So first, we have Iman GraphNet without alignment. So we took out the alignment step. So we don't have any alignment. So you can see we have a large error in MAE. The MAE is between the original ground truth functional matrix and the predicted matrix, the graph matrix, the generated one. And this is the MAE in closeness centrality, betweenness centrality, and eigenvector centrality. These are centrality measures to evaluate the topological soundness of the predicted graphs or generated graphs. So now when we use statistical alignment, so basically we took the block B, A, and we removed it with a handmade statistical alignment, with not in a learnable way, because here we co-learn, we co-optimize different losses. So when we did the pre-statistical alignment, we notice a nice drop in the error. So the error is decreasing, and we simply align the means and the standard deviations of the source towards the target. So it's very, it's a simple shift that we did to the weights of the graph edges. Now, Iman GraphNet, the green one is Iman GraphNet without any without the PCC loss, which is Pearson correlation loss. It's an ablated version, and this is topological loss. So we had different sub losses added up, and this is our final method. And you can see that using a two-tailed parity test, we got a statistically significant decrease across all these evaluation metrics, and we did some additional clinical inspection of the top discriminative connectivity. So the results were quite promising for a first model, simply designed for a graph super resolution. Now, if you want to learn more about this, the full video is available. I think it's a 20-minute video, and the code also is available on GitHub. So now we're going to go to cross-time prediction. So this is a work that has been developed also by my undergraduate student, and it has been published in both MICAI workshops and IEEE TMI journal. So here I'm going to present the early kind of layout of this problem and solution. And the advanced version is proposed in this paper, so you can have a read at it if you're interested too. So what's the problem here? The problem is that we want to learn how to predict the changes in the brain structural connectivity or in the brain adjacency matrix or the graph over time. Okay, so we want to predict this trajectory and generate it from a single observation. So now there have been a few works. There has been a few works on this, and they primarily focus on training decentralized or independent models. So in this paper, in this work, we adopted a federated learning paradigm. And we also added the constraint of missing time points. So far, the existing solutions at the time, they did not handle the problem of missing time points. There were like quite few endeavors, but very limited in generalizability, and they weren't tailored for federated learning. So let me introduce to you the 4D Fed GNN, and we had the plus and the plus plus. So first, let's start with this figure. So here, I think this figure is very important because we can see the distribution of the data in terms of availability. So each matrix here, we have a matrix. The matrix, the rows denote the number of hospitals. So we have distributed data. Their hospitals are collecting longitudinal data, which means time-dependent data sets, time series. And at different time points, you can see that some hospitals do not have the data acquired. So the data at this second time point is missing here. So now what we have designed in our solution is an algorithm where our generative GNN model acts differently depending on the sequence that we have, okay? So we have 4 cases. If the next time point, the follow-up time point is available, like in this case, and in this case, then our GNN will act as a generator. So it has a generation mode. Now, if the next time point is missing, like for example, this is a catastrophic case where you have double zeros, you can have even the whole sequence missing here, except for the baseline observation. So when you have, the next point missing, which means you don't have data to train on, you don't have ground truth, then the generator, the graph, will act as a self-encoder. So a self-encoder, it means it will act as an identity map. It will just take X, pass it over through the network, which is an autoencoder, and produces X hat. So the problem here is that 2 consecutive time points, if it acts as a self-encoder, like in these 2 cases, you may ask or you may wonder, oh, how am I going to predict actually the changes in the network now that I'm passing the same observations, identity map? So I'm considering that 2 time points have the same adjacency matrix, which is not our ultimate objective. So here, the secret, it lies in the federation process. So federated learning allows you to learn without having data, which is actually very fascinating to me. So let me explain that. So here we start with what we call, so let me explain what federated learning is for those who are not familiar with that. So first we have different clients, we call them here hospitals. So each hospital will create or collect its own data, okay? So let's say here you have 2 classes, normal controls and patients with dementia. So each hospital will has its own distribution, its own data set, local data set, and they could be distributed in different parts of the world, okay, or across different continents. Now, in federated learning, each hospital will train its deep neural network. So let's say it will has its classifier parameters, W1, here W2 for the second network, 0, 1, 2, and N. And then because their knowledge is limited by their local data distribution, what they do, they send out the local weights of their models to the server. The server will aggregate all these weights, it will average them, and send them back to each hospital. Now, the hospital will continue its training with the new weights. Now, what happened here? What happened here is that, I'm just kind of pausing, what happened here actually, so I was thinking. What happened here is that, each hospital has taken some, has benefited from some implicit knowledge gathered by these other hospitals. So the implicit knowledge is in the expertise of the classifier here. So if you have 3 experts, 3 doctors in these 3 hospitals, their collective knowledge has been communicated implicitly to this hospital H1. So H1 is benefiting from all these, trained models on completely different distributions of data sets. So it's somehow, without having access to the data, it's having access to the model itself that was trained on that data. So somehow it's kind of, learning from multiple data sets without necessarily accessing the data. This is why we call it data preserving learning paradigm. Now, here, this is called Fed averaging. There is also what we call a pairwise GNN layer weights exchange, which means that the hospital will exchange the weights directly with other hospitals through the server. And there's also the daisy chaining. There are different ways of exchanging, aggregating weights. It's a big area of research. But so now going back to our story, so imagine that a hospital here has a particular, sequence or a particular time point. So now that we're doing this, aggregation, this hospital that didn't have had an identity, a self-encoder right here trained. Now this self-encoder of this poor hospital will get updated by the hospitals that do have an encoder generator trained between these two time points. So the weights of this expert will get averaged or regularized by the weights of other experts that have available time points. And this is how basically this identity map changes during the training, because every time the server broadcasts the average model back to the hospitals, there is an update of the parameters, and the hospital with an existing time points is learning how to map something, of meaning. It's learning new mappings, okay, without having access to the training, without having any training data. So here we proposed an ordered way of communicating or exchanging weights. So usually the hospitals that are have the most available data, that exchange between them, and then all the way down to the poorest hospital. So we don't want basically to start aggregating the hospital with lots of zeros, because if you do that, you're going to kind of overwrite the expertise or the knowledge or the weights of the hospitals that have data. So we need to be smart about our aggregation and federation strategy. And this was what was designed and innovated in this work. So that's the key, or the gist of the key idea, or the gist of it. So now we applied this to 113 subjects from the OASIS data set. We used 5-fold cross-validation. So in each run, 1 fold will be used for testing, and the remaining 4 folds will be each will be allocated to a local hospital. So we compared against the vanilla method, which is a 4D GNN without any federation. So just the local performance of each hospital, and then the federated version, and then advanced federated version with the ordering. So here you can see this is the prediction at time point T1 between the ground truth matrix and the predicted or generated matrix at time point T1 from T0. And using no federation at all, you can see that we have a very large error, the purple method with 4D GNN, which is a longitudinal time-dependent graph neural network, generative network. Now we have the pink one which is with the federation process, Fed averaging, without the ordering, the smart strategy. And then we have this light pink with our proposed method. Now at time point T2, we have an increase in the MAE, and that's normal because the further you go in time, the harder the prediction becomes. And we still have a good kind of decreasing trend across all hospitals, the 4 of them, each one of them. So here we used the recurrent brain graph mapper, RBGM, as an architecture for generating the graphs. And if you're interested, also the video and the code are available for 4D Fed GNN and plus, and we have plus also. So the data set is available. So we have organized also a Kaggle competition a few years ago. This was part of my undergraduate course that I taught at ITU. As part of the course, we had a Kaggle competition at the end where we published a paper, and this compares 20 different machine learning algorithms for predicting brain graph evolution over time. So no fancy DL, but still we got some promising results, and it was interesting to use very basic, simple methods, especially within a machine learning course. So to see the potential of these methodologies without going deep. So the competition data set and the codes are available also on GitHub. So now we get to the last penultimate part. I think we still have one point to cover. So the graph integration. So if you guys remember, we talked about generation. Now we're going to talk about integration, because these templates that we will generate, or we will learn from the integration of multimodal heterogeneous data sets, we can use them to do some generative learning. And I'm going to give you in one slide one example. So what's the problem that we're trying to solve here? So the problem goes like this. So we want to integrate a population of different of multi-view brain graphs or networks. So we have tensors here. A tensor represents a single brain. These are multiple types of connectivities, for example, functional, morphological, different types of connectivities, and we have multiple subjects. Now, the distribution of the pink is different from the green and the yellow. So we have completely different distributions across domains, and we have variability across subjects. So our goal is to generate or integrate this multi-view data into a single matrix, one matrix that we call the connectional brain template, the template of the population. Now, why CBTs are powerful? So CBTs are powerful tools for group comparison studies and discovering also biomarkers in atypical brains and populations. So let me explain that. So for example here, let's say that we have a healthy population of graphs, okay? So these graphs, these are different individuals or samples. You can see that there are some shared patterns, some shared connectivity types. So they're very similar or almost identical. And remember that your brain graph is a fingerprint. So we need to have typical variability, typical shared connectivity, and typical very variable connectivity. So this is what makes these brains different, but all of these variations or variances, they fall under the normalcy distribution. So all of them are normal, but they are different, okay? So once if we create an average here, the problem is that averaging these will get us, will get affected by outliers, and this is a single connectivity type. So averaging is not a good, actually, operation or a mathematical tool to integrate these guys. So here our goal is to kind of create the average of this set of graphs by normalizing the typical variability and keeping what is shared, capturing what is shared and normalizing what is the typically variable across these cohorts. So now let's say we get a fingerprint. So this is the CBT of a healthy population, and we do the same for a different population, a disordered population. And if you simply compare these graphs, you can see that this actually what marks the largest difference between the edge weights. So this is identified as a biomarker, potential biomarker, or discriminative connection. So here, instead of doing intensive statistical analysis or deep learning, classifying, feature extraction, feature, like selection, all these steps are reduced into a simple comparison of two brain matrices. You take the difference between them and you get your biomarkers, and this is the power of CBT. So what is a good CBT? A CBT, we defined it to be well-centered, so it needs to occupy the center of a population. It is also discriminative, which means it captures what is unique about the population, so discriminative kind of marking or marker connectivities. And if we can learn a CBT which satisfies these two properties, then we can fingerprint the population and do comparative analysis with other populations. So the first work that was that introduced the concept of a CBT learning using geometric deep learning or graph neural networks is Deep Graph Normalizer, GGN. It was also proposed by my undergrad student, Mustafa, who's now conducting his PhD in the US. So here, the problem that we try to solve is basically how to integrate a set of multi-view graph population networks to get a CBT. So we know the problem. So we formalize it as a GCN-based model. Also, GCNs are very popular. Actually, they're in ICLR 2023. They ranked fourth after deep learning, reinforcement learning, and representation learning. So they're becoming more and more popular, GCNs. And here in this example, let me explain how our GCN basically works and how it is trained. So the key idea is to get one tensor, so one training subject. So this is a training subject. It's our tensor, our input tensor of multi-view brain graphs. And since we didn't have any features assigned to the nodes of each brain region, biologically speaking, we didn't have any features. So we set them all to 1. So we had identity features for every single node here, I and J, in this matrix, okay? So the feature matrix was a bunch of 1s. Now, because GCN takes in 2 things, it takes a feature matrix and it takes a structure, a connectivity, a graph adjacency matrix. So here we fed this tensor to each GCN layer followed up by a ReLU to learn the embeddings of the nodes of the brain. Okay, so we have NR nodes, and these nodes represent basically the graph nodes, right? So we do feed these edge attributes to consecutive GCN layers. So we have 3, and towards the very end of this, basically passing of the tensor throughout the network, we get the embeddings. So once we get the embeddings here, V1, V2, etc., of different brain graph nodes, now we can generate the CBT. So how do we do that? We apply a pairwise relationship. So in this example, we took, for example, the absolute difference, because that's how the original data was created. If the original data was created using Pearson correlation, you can use Pearson correlation between node embeddings, V3 and V1. Pearson correlation, you put a value here on this edge. So that's how we got this CS, this matrix for training subject S. Now you may ask me, okay, you passed this tensor throughout GCN layers, you got the embeddings, but actually, how did you get the center? How can you make sure that this, CBT here is kind of centered, is bringing each subject towards a center, right? It's centering the subject. So it's thanks to our loss. So the whole secret lies in the loss function that we propose. It's called subject normalization loss, the SNL loss. So basically, for every single training subject, CS, we take this matrix, which is right here, and we calculate its distance to a random set of tensors. So we vary here the views. So we have NV views. And we try to minimize the distance. So if you want to minimize the distance between this point here and all these colored, matrices, it means that I'm bringing this point towards the center of this manifold or this space, right? So basically, through this optimization, in each epoch, we are bringing CS towards, a randomly selected subset of subjects from the training data to do the centering. So imagine this. So what we do is, like, for every time we take a subject, and you have a cloud of points. If you imagine a cloud of points, every time we're bringing each point towards a global unknown center of the population. Okay, so we're collapsing a cloud of points into a single space where everybody becomes close. And this is done after training on all subjects. So you loop over all subjects. So you're going to get C1 for subject 1, 2, and training subjects. And all these guys will satisfy this equation, which means they will all be close to all the training tensors, which implies that they are all close to one another. So now we can apply a simple operation. So we can average, or we can take the median, because they're all at the vicinity of one another. So a linear operation will be reasonable at this stage. So this is what we did, and we get the final population CBT, which is the CREF here, refined one. So we tested this on several data sets, and this has been also extended into MGNNet, which has outperformed DGN, and we have dual high net and a lot of other variants that we published in the past 2 years that are not reported here, but this is the first model and we build on top of this. So we had AD LMCI, Alzheimer's disease, late mild cognitive impairment, with 4 different views, 6-dimensional, 6 frontal views in the tensor here, normal control and autism spectrum disorder. We did centeredness evaluation, discriminativeness also. We evaluated the quality of the biomarkers. So let me explain here. So for example, let's say we get the CBT of population A. CBT of population B. By calculating the absolute difference between these two matrices and summing over the columns, we can get the regions with the highest, alphas, which are the highest differences, and these will define our biomarkers. So that's how we find them. Now, what we did, we used a double-blind ML method, like multiple kernel learning, and we trained it independently, and we identified the list. We found out the list of biomarkers, and we looked at the overlap between the consistency between our CBT-based biomarkers, which is based on this simple absolute difference, and the machine learning biomarkers. And we found that we had a high reproducibility rate of biomarkers, which is hard to strike, especially even if you're training the same ML method multiple times, you may end up with different rankings of your features. So here we had up to 70%, sometimes more. So that shows that our biomarkers are kind of, are reliable because we use multiple experts to double check our knowledge, okay? So yeah, so we identify different biomarkers. This is a neuroscientific part, so I'm gonna skip through this for different cohorts and data sets and diseases. And if you're interested in learning more about multi-graph integration in general using GNN models and other models, we have published in 2023 a comparative survey, which is extensive. I think it's about 50 pages or more, with comparing all the state-of-the-art methods, including a Nature methods published in 2014. And since then, there weren't any publications on this. So we kind of scrutinized the literature. But if you find out any publication on multi-graph integration or graph integration after this one, please let me know, drop me an email. I don't want to miss landmark works. But anyways, so we built on that. And in this survey that was tested on, I think, 6 data sets with more than 30 evaluation experiments, DGN outperformed all of them consistently. So it shows the potential of generative graph neural networks in generalizability, reproducibility, and also accuracy and performance. So yeah, so these are the experiments we have ran. So the paper is here. Also the GitHub repo has all the Jupyter notebooks that generate all the beautiful plots in the paper. So if you're interested in reproducing similar plots, our codes are available. Yes, I won't go into the details. These are all properties of graphs in general, integration tests, segregation, hubness, centeredness, etc. So yeah, so now CBTs are very powerful. I just wanted to give you an idea about these templates that we derive from populations, and we can use them for so many tasks. And I have different projects on this topic. So one of them is graph super resolution, graph registration using templates to register a graph, an input graph onto a target graph, graph distillation, which means collapsing a set of graphs, a population graphs, onto a single fingerprint and doing learning on one-shot learning on one graph. Federated learning, we can use federated learning to do integration of graphs to learn the CBT in a federated way. So there are so many things, but let me show you an example. So this is a recent paper. Actually, it's not that recent. The topic is still ongoing, but yeah. So this is one of the first works that use templates for within a one-shot kind of learning, as a one-shot learning sample for training networks to do super resolution. So we start off with a low resolution graph, and our goal is to predict a high resolution graph in a completely different domain. Okay, so that's our goal. So the question that we ask here, can we lighten up the training of a GNN by leveraging representative data templates, which means, can we distill a large data set of graphs into a single one? So instead of training on all, we train on one. And let's see if that one, if it's so special, can it produce very good results? So now we proposed here TISNet framework, and TISNet is composed of different stages, but in a nutshell, we pre-cluster the data to disentangle heterogeneity. So we have different clusters. We apply DGN for each cluster. So we get a template, a cluster-specific template. And then we apply DGN hierarchically again to get the final CBT, the global CBT for the source domain. And we have the global CBT for the target modality or domain. Now we train a neural network F using only one single sample, which is the CBT, okay? The template of the source towards the template of the target. We minimize the loss, it trains fast, and once we see that it converges, we test our model. So let me show you the experiments here. So we use the similar data set, the SLIM data set, threefold cross-validation. We use similar for clustering. And here are the results. So first, we used, so these are the MAE between the predicted super result graph and the ground truth one, okay? Mean absolute error. So using a random one shot, which means we leave one out, but in a reverse way. So every time we train on one subject and we leave the rest for testing. So training on one single sample from our 155 subjects, I think. So we try to see if we can train on one random sample and still get good results. So this is the error that we started with. Now, once we do integration, interestingly, here with DGN, we get a decrease in the error. So at least across 3 evaluation metrics, topological, 2 difficult ones, actually between the centrality and eigenvector. Page rank is almost the same. MAE is in the connectivity themselves. So this is the most important one. Now, later on, we do testnet without these are the ablated versions without Pearson correlation and the L1 loss that we introduced. We see some good trends, especially for the MAE here. Now, for some measures here, it's fluctuating, but for the final testnet with all the losses and the clustering based, we get a very good results here across almost all evaluation metrics, even this one. So it's better than this one. So that's the lowest error that we achieved. And if you guys look here for training on all, so this is the interesting finding, and we still haven't gotten back to this problem. I think it would be very interesting to investigate or examine this problem in a more theoretical way. So because look, when we train on all data, we don't have a big jump, right? Comparing to training on all and training on one, we brought down the error from 0.3 to 0.1. So it's not normalized, but it's a big drop. And here, this is considered as the best expected performance, okay, in terms of error, right? So for other evaluation metrics, we see that this doesn't generalize across all topological graph properties. So for example, for between the centrality and eigenvector centrality, when training on all, we get really bad error here, okay? So training on CBT is the best, on one single template achieves the best results for this evaluation measure. So we don't have generalizability or consistency across all evaluation measures when we train on all samples, which is costly and requires a lot of resources. So training on one is always kind of affordable compared to this. So yeah, so this was I would say a prime work on this topic, and we're trying to extend this in different directions and lay down a more foundational understanding of this template-based learning. A last example here before I wrap up in a minute is the second example where we do graph-based registration. So here the goal is that can we boost the diagnosis of brain graphs or classification by registering or pre-registering them to a fixed template. So you can think about it as a regularization or a normalization problem. So we have an input network, and what we try to do is basically to pass this through a registration network that registers the distribution of the input network towards the distribution of the template. That is a prior knowledge that we have. It's a given knowledge. So given a fixed template T, we register every single training sample towards T. And when we do that, we get the green one. So this is the green registered sample. And we pass on this sample to train different classifier networks like GUNet, diffpool, GAT, etc. And we have shown in this paper published in 2023 that using graph registration network, we can boost the performance of GNN classifiers across different learning tasks using this registration template-based registration process. And this is because the registration normalizes the shared connectivities and it enhances what is unique about the sample, about the graph itself, and that allows us to discriminate better. So that's the underlying hypothesis. Now, yeah, so both paper and also codes are available. And just to kind of wrap up this talk, so there are several new concepts probing the field of generative neuro AI or generative GNNs in general. And so here are a few open questions: how to design accurate and budget-friendly generative GNNs in connectomics, and beyond that, not just in connectomics. So generative across domains, time, scale. We want them to be budget-friendly, which means we want them to be affordable to train, not to consume a lot of RAM or a lot of time and resources. How to super resolve brain graphs. So our Iman GraphNet, the super resolution graph model, was very, I would say, preliminary. But we can think about how to generate new edges and new nodes in a more kind of creative way and robust way. Affordable template-based GNN training. So we looked at the power of templates in solving complex learning tasks such as cross-domain graph super resolution. So to which extent we can leverage the CBT or the template-based learning to train these models and still have a good performance of these GNN models, generative models, and a good performance and a good accuracy. And if you think about LLMs, they are not affordable at all. So here, one of the areas that I'm working on right now, and I'm so excited about, is how to make these models less hungry, not for data, for parameters, for compute power, and can we strike a good, a good performance that is quite reliable compared to these huge models. So, and this is an open problem. There are so many, I think there are different endeavors or different leads trying to kind of reach to a good solution. But I think we need to revolutionize the way we think about deep learning in general and GPUs and stuff like that. So how can we also, can we learn how to generate data without training data? The answer is yes, if we can federate intelligently. And this is what I showed you earlier with the client-based learning or hospital-based learning that has learning from data with varying time series or sequences. So you don't have consistency in time or acquisitions. Yeah, so that's that. Thank you so much. And I'm happy to take your questions here.
All right, thank you very much. Thank you.
Thank you.
Questions?
I think, yeah, hi.
No, yeah, please go ahead. Yeah.
May I?
Yeah, please. Yeah. Hi.
Okay. Since this is a seminar that about machine learning and data dynamical systems, I want to refer to the very beginning of this tour de force survey that so nicely you described.
Mm-hmm.
And at the very beginning, not at the very, very beginning, maybe 10, 20% into the talk, had some evolution predictive
yes.
graphs. And my curiosity will be, how can this be compared to dynamical system differential equations approach?
That's a very good question. That's an excellent question. So, so I worked on PDEs before, so I'm not an expert in that area, to be honest. But one way is I was thinking about at some point, so let me just share the screen back. So it would be a very interesting problem to solve, because look here in the time dependent, so here the FT, okay? So this mapping, we can learn if we have an equation that can model these changes and we can express it mathematically, stochastically, or even, determine.
Yeah, if I may, actually, there is actually a way which is like through image registration, and yeah. So Tanya, are you here or no? Yeah, I see Tanya Shma. I mean, she's like.
Yes, I'm here.
Yeah, okay, exactly. So I think this is where you could answer, right?
Yeah. So.
Yeah. Okay. Sorry for interrupting, Islam, but there is like we have a way actually in dynamical systems that are called like image registration, and then your FT could be where we could use Hamiltonian dynamics, and so.
So is that true? Is this through geodesic shooting? Because I use this.
Yeah, exactly.
Geometry, and transport. So so parallel transport. So I think I've done that. I've done this a long time ago during my PhD. So I used diffeomorphisms, and I used also varifolds, and at the time it was about morphing or like registering, evolving these shapes. They have these kind of, it's using geodesic shooting. So that's another alternative. Now, the thing about this solution is that it is really time consuming to optimize. So I've worked on that. So it takes a lot of time to solve these equations, especially if you have meshes with thousands and thousands of nodes. So maybe for images, I would like to hear more about those who work on that, especially these days, because I haven't gotten back to that.
I had a discussion with Tanya, who's here, and a colleague, Human Ohadi, who developed like a kernel-based method where you actually use less data, but then you have like a dynamical system that evolves. But you use like much less data than than maybe, I don't know what you did like in your PhD, but it seems to be consistent with that.
So what do you mean by using less data? So basically, you're trying to kind of, is it for simulating, or is it just to partially solve the problem, and then you're doing a hybrid approach that marries AI with, because you can train AI models, deep learning models, to derive the parameters of the geodesic shooting. So I had a paper published last year also on that. So it tried to kind of predict the deformation by simulating, these equations. So you solve the dynamical system for many samples. You build your training data with all these equations solved with all the parameters, and then you give it an input sample, and then you try to derive the parameters without solving the equation itself, and then you just do the simulation and you predict. Is that something similar to that?
I think it's something similar to that, but we will not be using like a neural network. We'll be using kernel methods that are like more rigorous, mathematically speaking. And then based on the kernel, you can build a neural network by taking the inner product of the feature maps.
Oh, nice. Okay.
So that's maybe where Tanya would talk if she's around.
Very cool. Awesome. I'm taking
my question.
Yeah.
So this sequence that you show here, is it like analogous or similar to a convergence process in iterative way that people do in classical numerical analysis?
I'm not sure I'm getting the question, sorry.
Well,
so I think you're talking from a different field. So can you maybe reword it?
This is a sequence of graphs, right?
Yes, it's a sequence of graphs,
And when I improve a method, when I improve a solution or a process in numerical analysis, I aim at convergence at the optimum or at the real solution.
Yes.
So have we here on the rightmost panel arrived at convergence?
Yes, we have. We have very good, in terms of, it depends how, so it depends on how you define convergence. So let me explain the way I understand it using my AI terms, okay? So basically what we have, so when you say, for example, this is a ground truth observations, this is what I observe. Do I pass through that point accurately using these generative models? So if you say the model converges or approximates very well the current ground truth observations at this time point, the answer is yes. So if that's what you mean. If you mean something different in terms of mathematical properties, so I may not be able to answer that. But what you intend by your question is that, do we actually, does this FT meet, like the generative model, does it approximate very well or get closer to the ground truth trajectory of the ground truth observations of our training data, because we have ground truth there. So we're trying to approximate what we see through the training and optimizing the loss. Then my answer is yes.
Okay, wonderful. One more comment or another curiosity.
Yeah, so sorry, I think that Tanya wanted to answer maybe, and then you can ask your question again, Itamar, but I just want to have Tanya's feedback.
Oh, okay. Thank you very much. Hello.
Hi.
Thank you for this interesting talk. I did miss part of it at the beginning, but let me clarify my understanding, which I think might help someone else, which is that the time in this diagram that you're showing right now, f of t, that
Yeah.
is physical time, and we're talking about real data that exists at multiple time points.
Exactly. Yes.
So that's very different, Boumediene, from the situation where, where we're doing image registration and we have this sort of fictional time that's being used to shoot along a geodesic to get.
I see.
Yeah.
Right.
Yeah.
So you
Okay,
can.
this is something I didn't catch. Okay.
Right. So you can use geodesic shooting to do regression for a sequence like this, and that has been done by some people. I can't remember who.
Joneman, Younes, I've read all their works, the early guys, the landmark guys.
Yeah, so it is relevant, but not quite in the way you suggested. And I think it also relates to the question we just had, and you already clarified that very well, Dr. Rekik, that the convergence you have in mind here is convergence to one of these 4 time points that's showing. It's not convergence as t increases, right?
Actually, to all of them as t increases.
Oh, I'm sorry. Okay,
so we hit all the points. So
all the points, but when you're talking about convergence, I thought you were talking about like convergence of your machine learning method over the course of training, so that you could match each of these points.
Yeah, so that's right. So you could match each of these points. So each point, yeah, that's right, yeah.
Right, but it's not that you're trying to match one thing, and as t increases, your match gets better and better. In fact, it's
no, that's not true.
As t goes on, your matching is not as good because it's harder to predict into the future. That's all I wanted to add. Thank you.
Yeah, thank you so much for the comment. Thank you for the great question. Thank you.
Yeah, that's a good clarification.
Yeah, actually, I had one question out of curiosity I wanted to ask you. So,
yeah, sure.
First of all, I want to thank you for your amazing work, as I've told you at the beginning of this presentation only. So, I had one curiosity. I just wanted to know that from your work, is there any way we can predict the intelligence of any human being from these works that we are doing? Because as I know that currently scientists have now been able to give a proper answer to this question, that whether we can predict the intelligence of any person from I think that it is one of the biggest question which is still left unsolved. So I think that is there any way so we can measure or compute or by some way we can predict the intelligence from these graphs?
That's a very good question. There are lots of works on this topic, but very reductionist, I would say, and their definition of intelligence. So, for example, there are a few studies that aimed at predicting the IQ, the verbal intelligence score, VIQ, the fluid intelligence score, FIQ, from brain graphs or brain connectomes. But these are very reductionist measures of what defines intelligence. However, having said that, even starting out with these measures, they were able to predict quite well these values for large scale populations or cohorts of individuals. So we worked on that. Actually, we designed regression, I think it's called reg GNN or something, or RGNN, where we regressed out a graph onto a single score or multi-scores, and each score represents a measurement or a quantification of the intelligence of the input graph matrix or individual. But your question is really, really cool, because I think, it's a bigger problem, and it depends on how we define intelligence. So these models, they just take our definitions. But if you ask me, can we leverage the power of AI to redefine some aspects of intelligence for us? That would be a great question to answer through AI, because we won't have consensus on that easily. Yeah.
So I had just one other curiosity, that is the current slide that was just presenting. I am just going through the image, that is the brain graph evolution prediction, the trajectory. So I was just curious, this question is also related to my last question only, that is there any way, so suppose we take the graph of the brain of a human child who has been newborn child, and then as we move forward and then as the child grows older, then
Mhm.
can we somehow predict that whether the child is getting, means his intelligence is either increasing, there is a child is getting intelligent,
Yeah.
or it is
Yeah.
getting dull or something
Yeah, so I worked on
symptoms?
during yeah, that's a very good question. So we still have a few ongoing projects on the baby
Yeah.
connectome, using the baby connectome data set that was collected at UNC, University of North Carolina.
I
And
think that this somehow relates to one of the previous slides that I think that was just shown. It is somehow look like the heat map one that was just
yeah,
showed in your slides. The age
yeah.
was just 0, then 3 years, then
Yes.
75 years,
So
something like that. Yeah.
yeah, very good question. So I
Yeah,
think
yeah.
we had some preliminary work. Actually, we worked, we had several publications and a lot of different deep learning and AI models that did this prediction very well. We solved it. So we can predict for typical populations, but these models are still very simplistic because first they're trained on one cohort. The cohort is limited in number, so which means we have only, I don't know, like 500 subjects or even less. So if we are able to predict these patterns, it's still, this model is still very reductionist because we don't have founders, we don't have any causality, we don't have epigenetics. So it's all the setting of the whole problem is reductionist from the beginning. But still, even with that, we are able to approximate or to predict this 3 month from this 0 month at especially the most discriminative and important changes. Now, there have been also several studies which predict, given a certain time point t0, we predict if this baby is at risk or infant is at risk of developing schizophrenia. And these depend on data collection. So it depends a lot on the data. So if you have a lot of data, you know, like time series or like longitudinal data following through infants or neonatal
I think that this somehow related to forecasting, right? If we can
exactly, it
analogous
is
to the
forecasting.
forecasting time series here.
Yeah, so I think the forecasting for healthy small cohort, it works, but if you want to generalize to other cohorts for different distributions and different properties or from different continents, different genetics, maybe it won't be able to kind of generalize well. And this is a problem prominent everywhere, not just in baby development. But I would say there's a lot of potential. And if we integrate, for example, causality and we integrate other variables, we will be able to sorry, we will be able to predict, I think, well. But yeah, so you always start with the reductionist solution, and then you want to add more layers and more variables in your modeling along the way. And hopefully with that, we will be able to, for example, predict the different things or different questions that we will be asking the data at time point t0.
I see, yeah.
So,
Yeah,
Claudio,
thank you for answering my questions. Yeah, thank you.
Yeah. All right, thank you.
Thank
Claudio,
you.
do you want to unmute?
Yeah.
There's a question on the chat.
Yeah.
Yes, so that's a very good question. So there is a question, yeah. So okay, Claudio is asking, awesome. Sorry for missing. Okay, so could it be possible to achieve reliable results using small samples and 8 individuals with presented approaches? I think yes. So here's the thing. So the way I would answer this, it depends on the diversity of your samples. Okay, so imagine, so let me go back to this figure so you'll understand. So you need to quantify the diversity of your samples. If you have samples that are very similar, they will map onto you know, like the same point, which means you will not have representativeness in your data. And if you don't have variance or representativeness in your data, then these 8 will not count. So for example, look here, you can have millions of samples in this cluster, okay? So millions or billions of samples, but because all of them are very close to one another, they would represent, they don't have diversity. So these billions of samples, because they are very similar and they lack diversity, they won't contribute a lot to your training because there's a lot of redundancy. So what you want to have, even if you have 8 samples, you want them to be diverse, which means you want them to be located in different, you know, spread out on your manifold. So they don't collapse to the same representation subspace. So you have diversity. And with that diversity, you can easily, I would say, do classification. But this is a very vague question. I don't have a theoretical answer to that or a very kind of a fully grounded answer. But I would say that sometimes training on a few is better than training on many. And you can get some reasonable preliminary results. So I won't deter you from training models on the small data set. Actually, you can back up your story, but a huge line of works on one-shot learning paradigm. So you can check one-shot learning from one. There's also a paper published in Europe a few years ago. It's called fewer is better or something. So you can try to find these papers and then yeah. So I wouldn't say no, but check the redundancy in your 8 samples.
All right, thank you.
So
Thank you.
I wanted to ask a question about the graph that you are generating.
Mhm.
Which model are you using to generate? Are you using diffusion model or?
No, not diffusion. Diffusion are very hard to train. We're using GCNs. Very simple, very effective. They work everywhere. GCN layers, we did add some attention, and they're easy to train. They're reproducible because we tried different models, baseline models with different types of convolutions. So we use GCN-based convolutions primarily, and these are, you can add, attention mechanism, you can add multiple blocks, multimodal fusion, you can play with that. But the message passing here is a bit simple because our data is simple. They're similar in structure. We don't have a lot of differences in structure, so we don't need highly expressive message passing and fancy stuff. But of course, if your data stipulates that, you can play with these and use more advanced message passing approaches in, for example, GCNs, or you can use diffusion models or transformer graphs. So it doesn't really matter. So this is kind of a model agnostic approach. So any generative model would work, but we use the baseline model using GCNs because throughout our experiments and different, through the exploratory stage steps, I think the other ones are hard to kind of tune. They have so many hyperparameters, especially for the diffusion-based models. Although they're so cool, but they're hard to tune pragmatically on data sets.
Please, I didn't get very well. So how do you define your model GCN, or it's based on what?
Oh, so let me explain here. So 1 second. So it's based on a graph convolutional layer. So it has a simple filter that you're learning, a mapping function for each node that is neighborhood that aggregates neighbors. So this is just a simple GCN layer. So for example, if you're not familiar with GCN, so the idea is very simple. You just apply convolution to each node of the graph. And then after that, you do some aggregation, and this allows you to aggregate embeddings through the local neighborhood of each node. And then you pass it on, you update the embedding of the current node using itself and its neighbors. So here, for example, in this Iman GraphNet, we use a graph convolutional layer, one layer, so one hop, and then a dropout layer we added there. And we have a batch normalization layer just to control the variance of the learned embeddings between different layers and avoid, gradient chattering and some funky behaviors of the loss training. Yeah. So it's very simple. So think about it. This is similar to CNN on images. We just replaced a CNN layer with a GCN because we're working on graphs.
Okay, thanks. I asked this question because I'm working on knowledge graph, and
Yes.
we're working on knowledge graph generations.
Oh, nice.
Yeah,
That's
so,
advanced. Yeah. That's, I think, more difficult. Definitely.
Yeah. Okay,
So,
thanks.
Cool. Thank you so much. Thank you.
May I?
Yeah, sure. Thank you. Yeah.
Yeah, I have 2 comments. One is, since you mentioned those differential equations difficulty, that different root, I wonder if you're familiar with the work of T1-w in Duke University, who did multi images processing, and he's also using AI. That's
nice.
one.
What's the name?
The
Sorry,
other one,
you can write it later on. Yeah, thank you.
The other one is few years ago, because you were talking about baby and intelligence. few years ago, I suggested to some psychologists and other people a project that maybe your methodology can resolve. And the project to be shared here in public is, can we learn the wiring of the brain as a function of mother tongue, namely a study of different mother tongues, different languages, because we're doing LLM these days, and that all connects together.
That's a very good,
Because it will set
very interesting problem. Very
the
interesting
weights
question.
in the graphs will be set by initial connotation or utterance of mother tongue and facial expressions, etc. So I'm very excited to hear about that, if you have data sets of that, or one of your students will apply it, or any one of the listeners here. I'm not in the doing mode.
Unfortunately, I think this is a very interesting question to a problem to kind of address. How does I'm sure there are many scientific neuroscience or studies on studying or comparing how the brain networks of statistically matched cohorts in age and sex. They differ when you come when your mother tongue changes. Perhaps there are some preliminary studies from a neuroscience, pure neuroscience perspective and medical imaging, but connecting that to AI would be something very interesting. So there is what we call a neurosymbolic AI. So I'm just Googling this. So neurosymbolic AI it integrates basically across all kinds of languages and reasoning and learning paradigms. So neurosymbolic, I have remember I asked a senior professor, Professor Rasul. She works on this topic, and it's first time I saw her talk. It was very fascinating to me because she told me basically what they're doing in neurosymbolic AI. They're learning a transcendental AI language that transcends all these kind of, the image language, the text language, the math language. So it's a bigger umbrella that unifies and looks at all these symbols and makes sense out of them, whether it's text, it's video, it's what else do we have? Lots of other modalities, math. So it can logic also. So neurosymbolic AI is the abstraction, it presents the abstraction of all these kind of data, and it is able to generalize across all data sets. So if you have a neurosymbolic deep learning model, it can learn from all of these. So that's the idea I got, if I'm right, if I'm not wrong. I'm not an expert in neurosymbolic AI, but I think this would...
Yeah, you're right. You're right. I
Since
agree.
We have a neurosymbolic AI person here. Cool.
Since you mentioned transcendental, I'm now going tangentially to meditation analysis.
Yeah.
Okay.
Yes.
And that's since we are doing all this brainwashing now. I mean, brainstorming, sorry.
It is washing and it is storming, both.
They may be close, they asymptotically touch. Anyway.
I recognize that. I love skepticism, so just shoot. Yeah, so you mentioned meditation. Sorry, can you continue your line of thought?
Well, in different cultures or different starting points, meditation effects may be different for different people.
Yes.
Okay, and again, a good study of that or good data of this may tell us much more about this mysterious process called meditation, and could be even go tangentially to dream analysis.
Yeah, I totally agree. There has been amazing, fascinating studies that I read a long time ago. Unfortunately, I didn't remember the name of the authors, but there was they discovered that the temporal lobe completely kind of switches off in subjects who are highly meditative, like they reach a highly meditative stage. So there was a group study, comparative group study, where they brain scanned people during in a meditative state, and then people who are in a regular state, and they discovered huge brain differences. So there are, if you think about it as meditation, yoga, prayer, you name it. So these things definitely change your brain. Even like there has been a study a while ago published in one of the top journals on how books remap your brain. So reading a book, even a novel, they did this study for, I think I forgot which university. I don't want to misspell, but it was in the
Or
US.
attending Zooms. Attending different Zooms may also have
Everything. If you think about it, everything changes your brain. Everything affects it, right? So it's a big delta or a small delta, and you build in your brain what you inhabit, the habits you inhabit, right? So if you repeat the same thing over and over again, it gets consolidated in your brain wiring. That's very kind of
It's like
so I wish we had neuroscientists here. So I'm not a neuroscientist.
Therapeutic dimension, one can go to self-hypnotism.
Oh wow!
I would love just to remind me of the book by Dodge, the brain that changes itself. So there were
yeah. I know. I read that book. That was one of my favorite books. Still one of my favorite books. Yeah, Norman George. Yeah.
Yes, I know him personally.
Oh, you do? Oh my God! I'm a big fan.
He's in my city, Toronto. So.
Oh, you are in Toronto right now?
Yes, I am.
Oh, I love Toronto. I love Toronto.
Okay, show up and let me know.
I would love to. I will let you know. Definitely. I'll take your contact from Boumediene. Definitely. It's a very interesting conversation. Thank you for sharing the thoughts.
We have wonderful restaurants.
Yeah. So Itamar has been like one of the first people who has been attending the seminars for the last 4 years or so. So he has been always here asking very good questions.
Very nice.
For your coming all this time, Itamar.
Thank you.
All right. So thank you, Islam, for your.
I just had another question. So out of curiosity,
sure, are you okay?
I just wanted to know that.
That's fine. I still have a few minutes. That's fine.
Yeah. I just want to know that as these days the suicide rates are getting much higher than the last decade, as you can see throughout the world, or at least actually I am from India, and in India the suicide rates are getting much more higher than the last decades. So I was thinking that as I remember that about the morphological brain network just look like the heat map according to me only. Just you have just shown. So I was just thinking that is there any way that we can predict that as a child who is born and as who is getting depressed for any certain reasons like that regarding any academics or any personal reasons, so whether he's getting a chances to get, means the suicide rate is getting higher in this brain or not, means is there any property that we can either a person who has committed suicide and a person who is getting depressed, and whether he can commit suicide or not, whether this can be judged through this graph convolution GNN or not. Is there any way? Because I think that these questions, and I think that if we can somehow try to solve these problems, then I think that it will be beneficial for the society, that it can be just that whether a person can commit suicide or not, actually, to some extent, if only possible.
Okay. So I don't think you can, there are things that are, so if you think about it a little bit logically, but maybe not. So if we try to simplify the problem, right? So suicidal thoughts, I don't think they will, it depends on where you get into that cycle. So depression doesn't set off like abruptly, right? It's a process. There are lots of things that are happening, and at some point you hit that irreversible biological state where you cannot go back unless you have medicine or you have things. So I'm not an expert, so I'm just, please do not take my words for, I'm not an expert. I'm just trying to think out loud, and I have some knowledge a little bit. So I think the key thing, if you catch yourself before you fall into that irreversible state or mode or space or whatever you call it, then you can always kind of slowly reverse back. Okay, so you can take yourself outside from it before it biologically changes your brains and your neurons and your wiring, okay? But once that sets off, once you hit that threshold, the whole system starts to kind of collapse and you have irreversibility. So you need to force that reversibility through medicine. So that's the idea. So you force the system to go back to the balanced state, or whatever you call it, that kind of nice state, right? Normal one. So if you ask me, can we predict so logically speaking, if you start to, if your algorithm can pin down subtle alterations, early markers of depression, I'm sure there's a lot of works in neuroscience on depression and imaging, a lot of cohorts, a lot of studies. So maybe you may want to read on those a bit more. But my understanding is that logically, if you can see that delta or spot that delta early in the graph, then you can predict that brain may experience depression after, you know, like you can have some prediction, okay? So you can train a GNN to predict the irreversible state. And what that requires, unfortunately, for now, is to have people scanned and tracked. So, and this is not good, which means we're going to track your brain constantly, right? So, okay, so for my health, maybe I want my brain to be, I have a digital twin. That's what we talk about these days, right? You're having a digital twin where we're tracking you from birth till death, and all of your data is on the cloud, your health, your brain wiring, your brain graph, your heartbeats, your blood pressure, you name it, okay? So I don't want to discuss that. That's a huge topic. It has pros and cons. But let's say that if you want to spot that using MRI, which is really heavy, I mean, it's not a micro MRI. You need to sit down in a scanner for an hour, and it's claustrophobic. So for some people, and doing that often, it requires a lot of energy. It's costly, so it's not affordable to do these things. So I think we need better ways to spot this early, not maybe using MRI, maybe using EEG is more affordable if we can spot these changes. Maybe using a headset that, you know, kind of captures a few signals and does some image processing and builds the brain wiring in a very reductionist way or simplistic way, but still can detect these changes early. Like, you know, we call it now, you have an earable, and it can tell you things about your breathing and your heart. And so maybe we can have an earable or whatever, or like something on top of your head, and it can tell us things about your brain. So this is something that would be a new technology. But even with that technology, I think you're talking more about predicting these the likelihood of a suicide or getting depressed, but I think we need to focus more on preventive medicine. So there is a whole lot of work and a lot of money going into fixing what has been broken, but we need to ask ourselves why the system has broken in the first step, right? So maybe trying to kind of do research on preventive strategies for new generations and for people everywhere. I think that's more effective than just, you know, of course, both have merits, right? So I'm not denying the importance of diagnosis.
Yeah,
I'm saying that preventive medicine is very underrated, but it's very important.
All right. Yeah. So thank you for answering my questions. Actually, I just entered an amazing talk today. Yeah,
exactly. So I think we start with the
long day. Thanks everybody for staying around.
Thank you.
Thank you.
Could you please reply to my mail, because I
invitation.
really wanted to collaborate with you.